Good afternoon, everyone. Welcome to Connect Science. Today, we're very happy to have Brian Brown from ICON and Mount Sinai. Brian has been a pioneer in applying genomics and genetics to studying the immune response in cancer. He got his PhD in Canada at Queen's University and was a postdoc in Luigi Naldini's lab in Italy, where he published a really important paper defining new ways of regulating small RNA expression. Since he's been at Mount Sinai, is a professor there, runs their Genomics Institute, and has been really a pioneer in applying large data analysis to tumors and studying especially the importance of dendritic cells and tumor-associated macrophages in cancer. And today, he's going to tell us more about his work. I'd like to remind people that you can look up the seminars and recordings of these seminars on the website address given in this slide. If you have questions, we'll take them. Please jot them down in the Q&A and I will ask them at the end of the seminar. Brian, take it away.
Thank you. Thank you for the nice introduction, and thank you for inviting me. So I wanted to start off, before I discuss about spatial, one of the things that I think about and kind of what got us into the field and thinking. And, you know, one of the things that people don't realize or think about often is that even autoimmune responses are very spatial. And the reason why I think this is very important for tumor immunology in particular is that if you look, this is a disease, you know, vitiligo, alopecia, where you have an immune response which is killing melanocytes in this region of the tissue of the skin, but not right here. And you know, same here. You have cells in the hair follicle that are being killed by the immune system here, but for some reason the T cells don't move even a few microns, you know, a few cells nearby, and kill these other, you know, melanocytes in this case. And we don't know why. They're expressing the same antigen, but yet the T cells kill one cell and they don't kill another. And this is really, I want to highlight how spatial how spatial immune responses are. And, you know, this is a question we don't understand the answer to. So another example that people don't often realize is that diabetes has a similar kind of phenotype where, you know, it's not like the immune system goes in and clears all beta cells at once. We know this from projects like the NPOD project, where people die prematurely from diabetes, from other causes like a car accident. They look in their pancreas and they find that there will be regions of the pancreas where there are beta cells and then no beta cells, where the immune system has completely wiped them out. And again, we don't understand why is that localization, but it is. And this is telling you same antigen, but a different immune response just nearby. And so we have a story that came out that was published a few years ago by Judith Agudo, who is now a professor at Harvard, assistant professor at Harvard, where we looked at this type of thing where we took this mouse which expresses GFP in different stem cell populations. So it's the LGR5 GFP mouse, and they express GFP in the stem cells of the intestine, hair follicle, ovary, and mammary stem cells. And then Judith took either control T cells or these Jedi T cells that she had generated, which recognized GFP as their antigen, and she injected them. So the same antigen, but different cells. And when she looked in the gut at the stem cells, you can see that here's what happens when you inject the control T cells. The stem cells remain, they're all GFP positive, but in the mice that received the GFP specific T cells, you completely wipe them out. Now that was, you know, maybe expected. But then when you look in the hair follicle, we see something completely different, which was that the stem cells of the skin, which are GFP positive, they are not wiped out at all, and they remain even when you inject the Jedi T cells. Now, this is not I'm not trying to present this just to highlight Judith's beautiful data, again, which was published a few years ago, but just to highlight this fact that here we've injected all these antigen-specific T cells, and they kill one population in the same mouse, but not another population that are expressing their target antigen. Again, something is going on spatial here. And in this particular case, what Judith found is that when she looked at the expression of MHC class 1, and this is in the intestine, if you stain for class 1, just like all the textbooks say, all the cells will be class 1 positive, including the stem cells. But when she looked at the epidermis, you see lots of the cells are class 1 positive, but the cells that are GFP positive, which means they're the stem cell population, are class 1 negative. And you can see that here when she stains the skin. You can see that as soon as you get to the stem cell compartment, they've lost class 1. So here, and it turns out that the difference in this particular tissue really relates to the cycling nature of those stem cells. So when the stem cells are quiescent, they're not expressing class 1. But I again want to highlight this because it's showing you that there's a molecular difference between these cells. So you can have the same antigen, but then a different molecular state of those cells can define whether the immune system affects them or not, even the same animal and even in nearby cells. So what we were interested in studying, and I think it's an important area of the tumor immunology field, is how tumors control their microenvironment. Because really the goal of what we want to do, so of course we want to eradicate tumors, eat them by targeting neoantigens, but that may be limited to a lot of degree, and it may be limited so that, but we still, given what happens in autoimmune disease, it may be possible to take a self-antigen and turn it into an antigen that can be targeted by the immune system in a very local manner. And that is kind of the why we are interested in this question. And it's also an important question because we find that one of the most important determinants, one of the best correlates of whether a patient will respond to immune checkpoint blockade is how infiltrated by T cells their tumors are. And if you look here, it's quite remarkable. This is a melanoma. This is an image taken by my colleague, Miriam Murad and Sasha Nyadek. And you can see here in pink are the melanoma cells, and they're surrounded by macrophages in green. And then outside of the macrophages in purple and blue are the T cells and B cells. And looking at this pattern, it really is remarkable that this is orchestrated by the tumor. But you can kind of just by looking at this get an idea of how difficult it will be to wipe out these cancer cells if the T cells can't get in. And it should be pointed out that this is all the tumor. We would consider this still the tumor area. So it's not that the T cells haven't gotten in, they're just excluded from the cancer cells in this case. And so what we're trying to understand is what are the mechanisms of, or how exactly are the cancer cells controlling that? So my lab for many years, for I guess more than 10 years, has been using genomics type of functional genomics technology. We started out using microRNA and shRNA pooled screens, and the past couple of years we've started focusing on CRISPR or using CRISPR. And so when you do these type of screens, what you do is you take cancer cells or whatever cell type you're interested in, you take a library of vectors and you put them on the cells at low multiplicity of infection, and then you apply some selective pressure. It could be time or drug, and then you extract the DNA and you put it on a deep sequencer, and you ask the question, what is the frequency of the CRISPR guide RNA at before or after the selection? And from this, you can kind of infer what the knockout of the gene does. The problem with these screens, and they're extremely powerful, but they're really good for identifying gene functions related to cell fitness or things like apoptosis, proliferation. They're not great for identifying extracellular gene functions. In fact, for the most part, they can't do that at all. So they're really limited to screening for cell autonomous effects. So what do I mean by that? Well, if you think of there's two components to this. So one is the fact that when you extract the DNA and throw it on the sequencer, that's the only information you're going to find. So if a particular gene influenced whether T cells were infiltrating or there was B cells nearby, you cannot learn this information from this type of these type of pooled screens. But the other thing that people sometimes don't think about is that these screens don't assess entire classes of genes and that is genes that are working extracellularly. They can sometimes, but one of the problems here is that if a gene like a cytokine is being secreted, and if you knock out that cytokine in 1/1000th of one cell in every 1,000, then all the other neighboring cells are just going to produce that cytokine and compensate for the knockout of the cytokine in your one cell, and you'll lose your phenotype. So this sort of said to us, this is, we think, a problem. So we set out to try and solve it. And a few years ago, we had developed this technology that we call protein barcodes or procodes, in which we could barcode lentiviral vectors and resolve it at the protein level. So it's a very, very simple type of approach where you take a scaffold protein. We started with a transmembrane protein, NGFR, but mostly for the work I'm going to show, we use a nuclear localizing M-cherry, and you just fuse different linear epitopes to the scaffold protein. And this could be things like FLAG, HA, AU1, anything that can be recognized by an antibody. And then so with a limited set of tags, you can shuffle them around in every combination of 3, and you can get a higher order set of barcodes of, in this case, procodes. So if you have 14 different linear epitopes and arrange them in every combination of 3, you will get 364 unique proteins. And then you can build lentiviral vectors in which each vector has a different protein barcode and a different guide RNA. And you can now determine which guide RNA a cell is expressing by the combination of 3 tags they're expressing. So this was work published a couple of years ago. We can take, in this case, I think I'm showing you monocytes, put a library of 364 procodes into the cells. We can stain them for the 14 different epitopes, and then we can go to the cytoph along with staining with other markers, and we can resolve every population. And it works extremely effectively and actually quite cheap to do. But that's not what we were really ultimately interested in. What we really wanted to do was use the technology for carrying out these spatial screens. And so the way we do this is we can take those same cancer cells, we can put them, or to cancer cells, we can put a library of procode CRISPR vectors onto them, put them into a mouse, and then take out the tissue, and we can resolve where each procode is by staining them using multiplex imaging for each of the linear epitopes, and then determine by which epitopes the cell is expressing which procode and therefore which gene knockout they have. And the way we do this generally is using a multiplex imaging technique that my colleague Miriam and Sasha developed. But recently, and I'll show you just one slide at the end, we've been working with 2 scientists from Harvard, Sandra Satagata and Peter Sorger, to use this technique with cyclic immunofluorescence. I'll show you that at the end. So just as an example, we took 41 breast tumor cells, we transduced them with a library of 120 procodes, and then we injected them, and we take out the tumor and we perform this multiplex imaging. And you can see that you have here, where I'm actually only showing you 7 of the 10 epitopes because there's too many to look at. And you can actually see this patterning, which is really quite remarkable, of these cancer cells with all these different protein labels. So this is really a nice way to do clonal analysis. And what was kind of striking to us in this one was how heterogeneous it is. So you can see actually there's some structure here where you can see some regions of clonality. So in white here are the 41 cells expressing a particular protein barcode, but in general, it's actually quite heterogeneous, and I won't go too much into that. But now, when we took a different model, this is the model developed by Tyler Jax's lab. It has a KRAS G12D and a P53 knockout, and we transduce them with a library, same library of 120 procodes, and we inject them into mice. And then here's a one lobe of lung. We stain for this case 10 procode epitopes, and then you can look at it. And what you see here is that each of those, or most of those lesions, are actually extremely clonal, meaning they were formed by a single cancer cell, or single cancer cell, and you can see that here. Now, this starts to open up the opportunity for us to carry out really interesting screens where we can start to look at the tumor microenvironment of a single gene knockout. And it's kind of interesting because if you look around, you will see tumors that are very homogeneous, and then others where you see some heterogeneity. And I've had a lot of people ask me, well, maybe it's just, the difference between the 4T1 and the KP is just how you inject them. So one, you're doing sub-Q, in the mammary fat pad. and the other you're injecting IV. And it turns out that's not exactly true, because we took this other model. This is a pancreatic cancer cell model, KRAS G12D, P53 activating mutation. And when we inject those cells in matrigel into the pancreas, you actually start out by seeing a lot of heterogeneity. So a lot of these, I don't know, it's probably not so easy to see here, but you can see a lot of heterotypic clone tumor formation. And then if you look at later stages, you lose some of that. So something is happening over time, and you can see that even better here. So this is, the more yellow it is, the more heterotypic the interactions are, meaning that one cancer cell is sitting beside another cancer cell with a different procode beside it. But as you go look over time, you actually develop more homogeneity. And I think that's interesting to think about how this could happen, sometimes with the library, but sometimes even without a library, and think about fitness and even diversity in the cancer clones, and even maybe spatial, how they're competing in a spatial environment. But I'm not going to talk much about that. So what I'll tell you about today is some work that we recently published, where we took a library of different genes that we wanted to target. They had come out of some earlier work that we had done, and we built procode vectors to target each of these 35 genes. We introduced it into the KRAS, the KP cell, lung cancer model, and then we injected mice IV, and we performed our multiplex imaging. Even before we injected them, what we can do with the technology is we can actually phenotype the cells to determine the efficiency of knockout. So before we inject them, we transduce them with Cas9. And then in this particular case, we treated the cells with interferon gamma. So if you take KP cells in vitro and look at PD-L1, they don't express any. If you give them interferon gamma, they upregulate PD-L1. This is well known that it's one of the things that interferon gamma does. And this is our control right here. And you can see that this is the level of PD-L1 in cells that receive the control knockout. When you knock out any of these genes in the interferon gamma pathway, IRF1, JAK1, JAK2, STAT1, you can see that we completely lose the response to interferon gamma. You have no PD-L1 upregulation. And this is telling you how efficient this knockout is even before you inject. And the one I want to point out here is the SOX1 knockout, which you can see here. When you give interferon gamma, you actually get an upregulation, and that's because SOX1 is a negative regulator of JAK1, JAK2. So by knocking it out, the cells become more responsive to interferon gamma, and they upregulate PD-L1 about two to threefold more. And you can see other knockouts at a high efficiency, like CD47. And this is a really nice way to phenotype your cells and determine, how good your knockout is before you do those screens, which I think is extremely useful for understanding. And I actually will point out the STAT1 because I'm going to mention it later. The knockout was extremely efficient. So then this is what it looks like when we inject. As I showed you before, you have, sort of, this is one lobe of lung. You get many, obviously, from 5 from a mouse, and you have many mice. And you can see tumors all throughout. And then when you stain for the procodes, you can resolve which knockout is in which lesion here. And again, now what we are able to do, which we weren't before with traditional screening, is we can ask questions about how is this knockout changing the tumor microenvironment? And that's what I'm going to show you here. So before I show you that, what we can do, so the first thing we do is we basically segment the cells, and then we count. So how many cancer cells are expressing each procode, meaning how many cancer cells have a particular knockout. But then we can also ask other questions like how many tumors form. So not just how many cells there are, but how many actual tumors form, and then how big do those tumors get? And that's what this graph is showing here. So we're looking at what is the enrichment or de-enrichment of cancer cells carrying a particular knockout. As you can see, if we knock out CD47 or PD-L1, none of the cells, basically we get almost, I think out of 1,800 tumors, I think only 2 had these particular knockouts. But then what was kind of surprising to us when we did this, although I'll mention in a second, there's a literature on this, is that when we knocked out JAK2, when we knocked out IRF1, when we knocked out the interferon gamma receptor 2, those cells actually were de-enriched. And that was completely counter to what we had seen in vitro, where when you knock out interferon gamma receptor, the cells were actually no longer responsive to T cell immunity. But then when you, more responsive, but when you knocked it out, these cells actually didn't engraft very well, or they were cleared. And on the opposite end of the spectrum, SOX1, which is a negative regulator of this pathway, was actually enriched in these cells, which was quite surprising to us. And you can also see that the other one that I'll focus on here is SOX1 and TGF-beta R2. So when we knocked out TGF-beta R2, you can see that those cancer cells were also highly enriched. And then, as I said before, we not only look at what's the enrichment or de-enrichment of the cancer cells, but we can go a step further by saying, what about formed tumors? These are regions of clonality where you have more than 30 of a same procode in the same region, and then ask how big do they get? And those are a different type of question, because you're asking about questions of seeding and tumor formation and then tumor aggression. And those can give you, tell you something different about what the gene does. And, I want to say, we were surprised by these results. We were not the first to show this, though. There are people, Andy Min's group, I think, has done some of the nicest work. Also, Rafe Neumendorf at University of Colorado, Andy Min's at UPenn has done some really beautiful work showing that the interferon gamma pathway, when you knock it out, it actually can make the tumors more sensitive to immune clearance, depending on the antigenicity of the tumors. But what we were able to do then is take all those tumors, and I said before, from I think it was 11 mice, we had about 1,700 tumors. And not only could we look at enrichment or de-enrichment of a particular gene knockout, but we could also do things like pathology on the tumors. And so we had a pathologist blindly score all of the tumors. And this is what the control tumors look like. You can see these have a gene called factor VIII knocked out. It's not expressed in these cells. You can see this typical glandular structure of the adenocarcinoma. But then when the pathologist looked at the SOX1 knockout tumors, you see a very, very different structure. So they now look quite dedifferentiated. You lose the structure. They now are lipidic. And when we looked at the TGF-beta receptor 2 knockouts, these were really wild. They had this fibromucinous type of histopathology. You can see here a lot of mucin when we stain for it, and really a very, very different structure than the parent tumors. And again, you can do this at quite a large scale here. So what we really wanted to look at was how each of these gene knockouts influences immune infiltration into the tumor. So what we can do is not only do we stain for, in this case, 7 procode epitopes to resolve all 35 populations, but we can start staining for any other marker we want. And in this particular case, we stained for B cells, for CD4 and CD8 T cells, and then different myeloid markers, F480, CD11B, and CD11C, and then EpCAM to stain for the cancer cells. And in these radial plots that I'll show here, if you have a bar on the outside, that's telling you there's an enrichment of that marker positive cell. And if there's a bar on the inside, that tells you there's a de-enrichment of that marker positive cell. And you can see here that depending on the gene knockout, you get different tumor microenvironments. And the one that I'll point out here is the SOX1 and TGF-beta receptor 2. So those were the 2 tumors that grew the best. We had the biggest enrichment there, and yet they had very different tumor microenvironments. And the one that was really surprising to us was really the SOX1 knockout, where when we knocked it out, we see an enrichment of CD4 and CD8 T cells. And then when we look here in the TGF-beta receptor 2, we have a de-enrichment of CD4 and CD8 T cells. Actually, we didn't expect this either, but it's sort of what you would expect from a tumor that would grow really well, is that maybe they would be depleted of T cells, and that was actually the case, whereas this one, a bit surprising to us. So that's a measure of, what I just showed you was a measure of the T cells getting into the tumor, of what the region that we would consider the tumor. But if you remember, what I showed you with the melanoma is that, the T cells can get into a tumor, but sometimes they're excluded from the cancer cell. And that's actually a different measure than having has a different implication than tumors that are completely bereft of immune cells. So we can actually take our data and we can look at it in a different way. We can actually say, where in the tumor are those immune cells? And that's what these graphs are showing. So if you look here, when you look at CD8 T cells in the SOX1 tumor, it wasn't just that they had infiltrated in a larger degree, they had actually penetrated into the tumor core, and that's what you're seeing here. So again, surprising, because it wasn't just that they're bringing them there, but they're actually bringing them in contact with the cancer cells, and that wasn't happening with many other knockouts. So this is what the image is. You can go back and look at them, and here's an example of the TGF-beta receptor 2 knockout, where you can see it's beside a control and an MMP3 knockout, and you can see the T cells are much, much lower in number here than in the control tumors. And here's another example of that TGF-beta receptor 2 knockout. And then when you look in the SOX1 knockout tumor, you can see that you have T cells all throughout the tumor, as I showed you in those radial plots. Now, the question that we then want to ask, and I think this is an interesting one, which is how are neighboring tumors affecting each other? And I don't, this is a very unique question to that we can answer when we're doing these type of competitive screens in vivo. And I'm going to show you in a minute why I think it's relevant even to be thinking about and even to patients. But what we did is we asked the question, okay, if SOX1 are highly infiltrated, TGF-beta receptor 2 knockout are depleted or excluded of T cells, what happens when they neighbor another tumor? And I'll just show you this example. I won't go through what this heat map shows, but because I think it's a really remarkable thing to think about. And it comes back to, those images of the vitiligo and alopecia and type 1 diabetes patients, which is, if you look here, you can see in white are the SOX1 knockout tumors. And in this particular case, you have a SOX1 knockout tumor surrounding a TGF-beta receptor 2 knockout tumor. And in red and green are the T cells, are the CD8 and CD4 T cells. And if you look here, almost to the cell, you have a difference in immune infiltration. So the SOX1 tumor highly infiltrated, but that's not spilling in to the TGF-beta receptor 2 knockout tumor. These tumors remain excluded even when they're side by side with a SOX1 knockout tumor. And I think that this has implications for, potential pockets of resistance in patients. So if you think about, resistance mechanisms, we often, is it, one or two cells that survive? I think that's a very different thing, having, rare, infrequent cells survive against a targeted or chemotherapy. I think with an immunotherapy, if you think about pockets of resistance and where, you have many cells and they formed a different distinct tumor environment, that's a different type of resistance potential than a rare cell that resisting the immune system because of the challenge of the tumor to regrow that tumor microenvironment that's protective. And so I think this is interesting, and I'll show you why in a second. But this is work that's really nice paper. I think it's a really nice preprint that's out from Lucy Yates and Moritz Gerritz and others, where they're using this technique to resolve the genetics of patient tumors and try and look at subclonality spatially. And so here's an example of a tumor right here. All looks like one tumor, but when they use their technique they call basis, you can actually resolve that there's two different subclones side by side, and you can actually see that there's two different microenvironments that are distinct between these subclones. And so the reason I present this is only to say that there, sometimes when we look, or a pathologist looks at tumors and they see, a lot of heterogeneity, you'll see immune cells in one area, no immune cells in another, or fewer immune cells in another area of the tumor. It's kind of sometimes chalked up to kind of stochastic, these are just kind of random differences. But actually there may be an underlying genetic or transcriptional structure that's sort of driving that difference in immune composition. And I think that's something that we kind of want to try and resolve with this perturbed map technique we've developed. And I think it's going to come out from the different profiling studies like this one in patients. And so, why is that important? Well, here's an example. So we kind of reasoned that the SOX1 tumor was highly infiltrated. I showed you before that when you knock out SOX1, you get more PD-L1. And then, as I said, there was work by Andy Min and Rafe Newmandorf showing that, that you can have differences in antigenicity can influence the response here. And so we decided to take SOX1 knockout tumors and we put a 1-to-1 mix with a control tumor. And then we treated them with either isotype control antibody or anti-PD-L1. And so if you treat the mice. So these are the mice received both cells in the exact same mice. There's several mice here. If you give anti-PD-L1, you reduce tumor growth. But then if you look at the proportion, so we took out those tissues and we stained them either for the ProCode, the SOX1 ProCode, or the Cas9 ProCode. So if you look in the mice that received the isotype control, just like we found in perturbed map, 70% of the tumor burden in those mice is the SOX1 knockout. But if we take mice that received anti-PD-L1, so they also received a 1-1 mix, and when we treated them with anti-PD-L1, now we flipped this. So now the majority of tumors are the control knockout and not the SOX1. So in fact, in 3 of the 7 mice, we couldn't find a single SOX1 knockout tumor, despite the fact that without the PD-L1, 70% of the tumors were. So this is telling you that even when you have these clones and they would be growing, side by side, give anti-PD-L1, because the SOX1 tumors were highly infiltrated, and probably this is suggesting or probably indicating that these tumors were really being protected by upregulating PD-L1. There's probably other mechanisms as well, but by giving anti-PD-L1, now that they've become more highly infiltrated, they're also more sensitive to getting anti-PD-L1 than the control tumors, and that's why you see this improved clearance here in this competitive model. This is just to show you that we were able to validate the TGF-beta receptor 2 knockout. This was a really interesting one for us. It hadn't been shown in lung cancer, this immune excluded phenotype of these tumors. So we were really fascinated by why knockout of the TGF-beta receptor would result in this excluded tumor. And just to show you here, even at the early time points, we already started to see more TGF-beta receptor 2 knockout lesions. By 14 days, it was really prominent, and then by 28 days as well. And even at 14 days after injecting those cells, you can see that they already had this fibromucinous pattern. And then really at 28 days, it was really quite extreme. And then, as I showed you, as we found with perturbed map, you can see that knockout of TGF-beta receptor 2 in the validated mouse models also results in T-cell exclusion compared to the control. So TGF-beta receptor 2 is not commonly mutated in lung cancer, but several groups, including one here, Charles Powell's group, has shown that it's very commonly downregulated at the transcriptional and protein level. In fact, many, so this is some of the staining they've done, and they find very little TGF-beta receptor 2 in some of these lung tumors that they looked at. So we were kind of interested in, why would knocking out this gene result in an immune excluded tumor? So the nice thing that we're able to do with the technique is we can take a slice of tissue and we could put it on a Visium slide, so to do spatial transcriptomics, and that's what we did here. And then we take the data from Visium, we did unbiased clustering, and just doing, a sort of 2 k-means clustering, you can see that the Visium is able to distinguish the lung tumor lesions versus the healthy lung. You can see that here. This actually completely matches up with the H&E section. And then we're able to detect the transcripts from the lentiviral vector that expresses the procodes and the guide RNA. Here, this is just matching which regions of the tumor express the WPRE, which is a post-transcriptional element on the end of the transcript. And then this is, again, unbiased clustering, but we've now labeled it. But the unbiased clustering sort of says, well, these regions of the tumor are similar and these regions are distinct. And when we looked at the regions which were clustering here in purple, they matched with exactly where the TGF-beta receptor 2 knockout were. So distinct spatial regions, but with the same transcriptional profile. And this was probably one of the most surprising results to us in the paper, which was that when we looked at the TGF-beta receptor 2 knockout tumor, so they don't have, don't express TGF-beta receptor 2, but the most upregulated pathway was the TGF-beta pathway. And so we sort of scratched our heads there and said, why would that be? We did this, we were like, well, maybe there's some mutant and it's hyperactivated. So we took the cancer cells that had the knockout or had the guide and the procode. We treat them with TGF-beta. If you do that to the control cells, they'll phosphorylate SMAD2 and 3. And you can see that here in this phosphoflow plot. but when we treated the knockouts with TGF-beta, you see no upregulation, telling us that this was really a bona fide knockout. So then Sam Rose is a computational expert who worked on this data. He did some analysis using a database actually developed there at Dana Farber, CytoSig, where he looked at the transcriptional profiles of cells that had been treated with different cytokines. And what he found was that this signature that we're seeing in these tumors was very consistent, or the most consistent was with lung fibroblasts treated with TGF-beta. So epithelial cells as well, but since we knew that those did not signal, what it looked like to us, and as I think very apparent by the genes, I'll show that in a minute, is that what's happening in those tumors is the TGF-beta signal is not coming from the cancer cells because they can't turn it on. It's coming from the fibroblasts. And so what you can see here, in fact, is that many of these genes that were upregulated, so if you look here, this is COL1A1, gene mostly expressed by fibroblasts, and you can see here that exactly in the regions where you have the TGF-beta receptor 2 knockout, you have much more expression of this gene, which is specifically induced by TGF-beta. And so what this was telling us is that without even increasing, and we have maybe a little bit of TGF-beta increasing, but what we think is happening here, and I think this is interesting, and it's I'll say why, but what we think is happening is that in normal context, you have in a tumor, majority of cells in the tumor are the cancer cells, sometimes macrophages, but majority are the cancer cells. They express lots of the TGF-beta receptor in this case. You have TGF-beta around. It's sitting in the ECM. It's latent, but it's still sitting in the. And whenever it's able to dissociate and become bioavailable, it will bind to its receptors. And in a normal tumor, a lot of those receptors will be expressed by cancer cells. But now, when you knock out the TGF-beta receptor, when you have without changing the amount or the concentration of TGF-beta, you now have more bioavailable TGF-beta to bind to the receptors on fibroblasts and immune cells. And so this is why we think we're seeing this activation, this TGF-beta signature on the fibroblasts. And we think it's probably happening in the immune cells early on, but we know from work by Shannon Turley and others, very nice work, that upregulation of TGF-beta creates this immunosuppressive environment. So this is known to be the case, and it's known that when this TGF-beta is working through activating fibroblasts. So what we think is happening is that we're similar to upregulation of TGF-beta by losing the receptor, increases its bioavailability, acts on the fibroblasts, activates them, and creates this immune environment. Fibromucinous excludes the T cells, probably a lot of physical exclusion. That's why even when they're near the SOX1 tumors, they're not getting in. And I think that this concept probably plays a role more than we realize. And actually, we took this concept from the regulatory T cell field, where we know that one of the things regulatory T cells do is they express the IL-2 receptor, and one of the things they do is outcompete effector T cells for IL-2. And I think that this may actually be happening more than we realize, and it's something I think to look at in terms of what happens when you mutate or downregulate receptors. Do you change the competition for their ligands within a very local space? And I think it's something worth exploring. All right, so with that, I'll just wrap up with 2 slides and then the acknowledgement. So we've done this approach, which we call perturbed map, in a number of different models. I showed you the lung tumor model. We've done it in breast cancer, in pancreatic cancer, and ovarian cancer. It works extremely well. It's pretty low cost technology to use, and I think it's extremely effective for particularly looking for genes that operate extracellularly. If that's your goal, then I think it's a really useful screening technology. And then just this last slide, I'll give a shout out to our collaborators, Sandro Santagata and Peter Sorger and Lindsay Browning at Harvard there, who have been establishing cyclic IF to detect the procodes, and they've done a really great job at doing this. And so if you're interested, you can talk to them as well about we're still in the early stages, but I think it's looking very promising. And with that, I'll thank the people who did the work. Really great lab. The work was mostly led by Maxime Denan, super talented postdoc who's now at ImmuneAI. and Sam Rose, an excellent graduate student who's now in Dana Farber's lab, Ola Brauska, who developed the original ProCodes, and then many of the other people in the lab who helped this work and other work, like Alessia Baccarini, Sebastian Nielsen, and Gurkan Malaglu, and then my very close collaborators, Miriam Murad and Sasha Yadek, Dana Farber for her help and supervising Sam, and all the other people who helped with some of the computational and pipelines, and Gurai Akturk for the pathology. And thank you again, Judy, and for inviting me. This is fantastic. Thank you.
Thank you, Brian, for a really nice talk. We have a couple of questions in the Q&A. People can ask more, and we have time, which is nice. So I'm going to take my prerogative as chair and ask a question. So I was wondering, I think it might be useful to look at specific knockouts co-injected in vivo. And the question was, do you ever see TGF? So if you could use the TGF beta receptor knockout as sort of a sensor for T cells that might be generated in another knockout that were so efficiently stimulated that they might.
Yeah.
Do you ever see that or not?
No. So far, this is a question that we're interested in for many different reasons, drug, all these type of things to look at. But in terms of how, if one tumor will educate another tumor, the cells in that tumor to be more, effective or aggressive, or this is something, we haven't looked enough, but this is something that we are really interested in. One thing that we're discovering that I find fascinating, I don't know, I don't think it's that obvious, but maybe it is, I don't know how local all these knockouts are, how local the effects are. So I know I keep stressing that, but I say that because, we've seen even with chemokines, I didn't show the data, but, we were expecting them to be like, okay, well, you knock out a chemokine, but, the tumor next door is just gonna, they're so far acting, it's gonna compensate, and it doesn't happen. And I think that tells us something about the biology. I always say, I think, probably biology would evolve, and I don't want to say it evolved this way, but probably biology evolved to be very local for feedback systems, right? So it wants to be able to send a feedback to say, stop sending me that signal. And, being too distal is probably not something there are examples like hormones and things, but certainly cytokines is something you want to be able to turn off quite locally. So you want them to interact and not dissipate too far, I think.
Great, thank you. So Jonathan Frieden has a question. It's, I think, a little bit related to what I asked. Do immune cells learn to recognize tumor cells in one clone that is perhaps T cell infiltrated, then migrate and recognize cells and clones that weren't infiltrated?
Yeah, we don't know the answer. I would love to know that. I think, we're doing this in cancer, but this is a question, I talked to John Harris about this in vitiligo. If, this is part of the education, one lesion is educating the T cells. Do they have to go back to the lymph nodes? These I think are interesting questions. We don't know the answer at all, actually.
Right. Okay, so there's a question from Keith Loeb, and he wants to know, is the mucin from the TGF-BR2 knockout immunosuppressive? And does it have increased TGF-beta? So is it sort of acting as the reservoir, maybe?
That's a great question. I don't know the answer to that, actually. If mucin is immunosuppressive, that seems like a cool, that maybe people have looked at that. I haven't looked at that. I like that. But the point about TGF-beta, it sits in the ECM. I'm not a TGF-beta expert by any means, but I know it does sit in the ECM, and probably there's some where it's now you have more ECM and maybe more TGF-beta and all these type of things that are happening in those tumors. So I don't have a good answer though.
Okay, well, I think that was a good question. And so Jonathan Licht says the KP53 RAS model develops a lot of intrinsic heterogeneity as tumors progress. How can you use this technology to map the immune consequences of increasing heterogeneity within subclones?
Yeah, I think that it's not something that we've done a lot of. You definitely, I mean, I really suggest anyone to do it because I think I think adding spatial is really, important thing to do. And, I just sort of touched on the pancreatic. What I'm fascinated by is, even without a perturbation, you'll see clones take over. And I don't understand, if it's like one becomes more metabolically fit and then the guy, because I would think like if they start growing together, even if one grew faster, the other guys would still kind of dot inside and still kind of live there, the other clones. And we don't see that. It's almost like they get selected out and cancer biologists could probably figure that out. I'm an immune, I always tell my lab when they discover things like that, like move on to something that we understand better.
But maybe you could do, sequencing of the clones that become dominant and, see whether there are other genes that are differentially expressed in the dominant clones versus, or even as that clone evolves in the lung, it might be interesting.
No, the answer is, I mean, the technology right now so we will be doing that. I mean, we're waiting for, Murfish and Cosmics and some of these techniques to, be at the state where we can do this. I will comment, because we do spatial transcriptomics, we use the Visium platform. One thing, and we sometimes think, well, why don't we just do that? Because you get a lot of information. And the short answer is, because of heterogeneity, you really need a large sample space when you start to do in vivo and when you start to look at all these tumors. So if anyone, in the audience and listening is thinking about doing this, the reason why we do whole slide scanning, we try and get a lot of tumors, we got a lot of space. And I think it's going to be important for spatial. I think getting a tiny region of a tumor and doing a lot of transcriptomics is, could be a recipe for, a lot of noise and, well, not just noise, but, there's going to be just kind of, I don't want to say just natural variability because something may be going on, but I think it's something to think about. So sample space is a very important thing, I think, in spatial.
Okay, and we have a question from Greg Baker. Thank you for the great talk, Dr. Brown. Wondering if you have begun looking at tumor diversity in immunocompromised mouse models like RAG, knockout, or NSG, or immunoproficient mice treated with immunodepleting antibodies to delineate tumor intrinsic fitness in response to gene knockout from tumor extrinsic immune editing.
Absolutely, yeah. So I didn't show it, but we almost always, we spend a lot of time setting up our library, making sure that the knockouts are really good. Not all of them, but we try and do as much as we can. And we like to do that. I would recommend to do that. So, because in screens, you get a lot of false negatives, right? Like you have no idea if your CRISPR even worked well. So, but better that, the better. And then what we do is we put them in any mouse. So we'll put them in a rag mouse. We'll treat them with, chemo. We'll treat them, and I that's the best thing about these screens. I mean, is that it's we're doing like 35 or 56 genes. That's not a lot. But then once we'll do like 3 time points. I mean, I think that's the power is you do, a small library and then lots of different conditions and then lots of phenotypic parameters. And I think that's a very powerful way to do these things. We also, I would just suggest to anyone out there, if you have a lot of cell lines, I think, we use CRISPR, but I actually think like a great way to do this would be just, because all we have here is a barcoding technology that we can resolve. So if someone has like a freezer full of cancer cell lines, just, put a pro code in each one. So you have like 50 different, melanoma lines from different patients, put them in a mouse and see how they compete with each other, where they localize. I mean, a lot like the, maps that Todd Golub has been generating and using DNA sequencing, but now you're able to do some of this imaging that I think would be useful.
Yeah, does anyone have any more questions for Brian? I guess that's it. I mean, we're doing some. We've been identifying genes that are edited in early tumorigenesis, and it might be really interesting to make barcodes of those because I think they're going to have different different mechanisms for suppressing tumor immunity.
Absolutely, yeah. Well, we're happy. They're all just so people know, they're all not free, but they're all available on AdGene, so anyone can go. It's quite. We don't get any money for them, but AdGene's great. So you can go and you can purchase them and you can try them. It's not that hard. I mean, the computation is hard. I don't do it, but people have figured that out. So I would suggest, try it. It's not so hard to do. Thank you again for inviting me.
Well, thank you for giving such a nice talk. Bye, everyone.
Bye, everyone.